Assalamu alaikum students. I am Miss Aisha Abid from Army Public School and College, Khanewal. I welcome you all in our class 9, subject biology, week 2 by APSEX online support program. Topic is 1 and your teacher is Miss Aisha Abid. Our topic today is factors affecting the rate of enzyme action objective. We will be able to understand all the factors which are affecting the rate of enzyme action, particularly temperature. First of all, we will know about what are those factors which affect the rate of enzyme actions. Enzymes are very sensitive to the environment in which they work. It means that enzymes, as we know, these are the proteins and they are very sensitive for the environment. Environment is giving the enzymes the temperature, the environment is allowing them to work in a particular pH. So all these factors, pH and temperature, they are very crucial for enzymes. Any factor that can change the chemistry or the shape of enzyme molecule can affect its activity. So the first factor of our today's lesson will be temperature. Temperature is, as we know, it's the degree of the hotness and coldness of a body. So any increase in temperature will speed up the enzyme catalyzed reaction. This is the rule. When we increase the reaction, our reaction rate will be increased automatically, but it will happen to a certain point. It means that every enzyme is having a particular value which is known as optimum temperature, which means that the specific temperature at which every enzyme works at its maximum rate is known as optimum temperature. For example, optimum temperature for maximum working speed of human or any warm-blooded animal is 37 degrees. So this is a very important definition. You have to learn it and remember the example of humans or any warm-blooded animals of 37 degrees as its optimum temperature. So what is the role of optimum temperature in the rate of enzyme activity? Let's explore. Enzymes work at its normal rate when temperature rises to a certain limit. When heat adds up or when we increase the temperature, it adds up in its activation energy and also provide kinetic energy for the molecules to move faster. Hence, the reaction rate is accelerated, or the word accelerated here means the reaction rate is increased with the increased speed. Now, what is enzyme denaturation? It is an important term, just like optimum temperature, which is a very important term of temperature. But the temperature, when the temperature is raised well above the optimum temperature, for example, in case of human, we learned that this is 37 degrees. When temperature is raised to 30, 40, 45, and then 50, heat energy is increased, the vibration of atoms is also increased. It means that the molecules which are making up the enzymes, what are those molecules, kids? Those molecules are amino acids. The kinetic energy of those molecules increase, they start to vibrate more frequently, and hence what happens, the hydrogen bond between the amino acid, they tends to break down, which will ultimately destroying the globular structure of enzymes. The key structure of enzyme activity is their globular structure. This is known as enzyme denaturation. When the structure is destroyed, obviously enzyme is destroyed and we cannot take up our chemical reaction. Let's have a look at this video so that you may understand your concept very clearly about denaturation of enzyme and how enzyme
The activity of proteins which is not optimal can affect protein digestion in the body. If we look in more detail, denaturation of enzymes and any other protein means that the shape of the enzyme has changed in some way, usually a permanent change. The activity of an enzyme depends heavily on its shape, particularly of the active site which binds the substrate or substrates. This change of structure means that the active site is not shaped for optimal rate of reaction. So for example, if the temperature of the environment is too high for the specific enzyme, it may become denatured. Typically, enzymes that operate in warm-blooded animals tend to have an optimal temperature of 37 degrees Celsius, which is body temperature. Then as the temperature rises above that, The rate of reaction rapidly falls because the active site has changed shape, whereas enzymes in bacteria that live in the thermal vents have a much higher optimal temperature. Now here comes the chemistry behind this. When temperature increases, the individual amino acids that make up the enzyme vibrate at a higher frequency. This results in breaking of hydrogen bonds between amino acids and different parts of the protein chain, leading to a change of shape. At even higher temperatures, bonds between individual amino acids called peptide acids can break, leading the protein to fragment.
So that was how the denaturation of enzyme would take place if we increase the temperature above a certain limit. Now we studied the effect of enzyme denaturation. What happens when the enzyme denaturate? It results in rapid decrease in rate of enzyme action, which means that enzyme action rate is very much decreased, so that substrates are not converted into the product, or enzyme activity may be blocked completely with the help of inhibitors. There's a brain teaser for you. If birds have high body temperature than humans, for example, which is more than 37 degrees, what happens to their body's enzymatic activity if they are given temperature of 37 degrees? Your answer is reaction will be slow. You have to explain why the reaction would go slow. Alright kids, write your answers in your notebooks and submit it as a part of your AIS assignment. Now let's have a recap of what we have studied in our today's lesson is the effect of temperature on the rate of enzyme action. We learned that how temperature is affecting the rate of enzyme action and how the rate at which the substrates are converted into a product with the help of particular enzyme. Then we learned the term enzyme denaturation. This is the term which is used for when the active site structure amino acids it is altered and the active site has changed its shape, which means that enzyme no longer recognize its substrate, hence substrate is not converted into product. It means that enzyme activity is blocked temporarily or permanently. Let's have a view of this video to be clear about the concept.
In our last video, we looked at why we need enzymes and how they work. In this video though, we'll consider how temperature and pH affect the functioning of enzymes. And in order to measure this, we need to know how to calculate the rate of a chemical reaction. Let's start with temperature. This here is a graph that shows how the rate of an enzyme-controlled reaction changes with temperature. We can see that as temperature increases, so does the rate. But after about 37 degrees, the rate starts to drop rapidly. This is because the high temperatures start to break some of the bonds, holding the enzymes together, and so the active site starts to change shape. If it changes shape too much, then the enzyme won't be able to bind the substrate and catalyze the reactions anymore. And at this point, we'd say that the enzyme has been denatured. The temperature at which an enzyme functions best is known as its optimum temperature. So the optimum temperature of this enzyme would be 37 degrees, because that's the temperature at which the rate peaks. As the human body is generally kept around 37 degrees, this is a pretty common optimum temperature for human enzymes. But different enzymes can have different optimum temperatures.
So that was all for today's lesson. I hope you understand and enjoy today's lesson very much. Don't forget to answer that brain teaser which I have provided you in our lesson, and complete your assignment in your due time. Thank you so much students for your attention, and stay healthy, stay curious. Allah.